This is the anatomy of the lymph node, right? So here you actually see a microscopy. All right, this is a dye microscopy of the lymph nodes. That's the true aspects of it. Again, lymph nodes are really tiny, guys. Right? How big are the lymph nodes? Anybody? It's less than a centimeter, guys. It's really, really, really small. Right? Of course, as they get inflamed, of course they're gonna get bigger, right? Because there's inflammation around it. But you know, we're talking about millimeters, so it's about five millimeters. So the next one is, so all lymph as it goes up towards the heart is going to enter the afferent vessels. So as lymph enters here, right, then this lymph is going to go through this structure. As it moves from the structure means what is the structure? So the structure is going to be the outer cortex. So as it moves through the outer cortex, we talked about the river and pebbles, right? So that fluid, that lymph fluid is going to move through the pebbles, and of course attached to these each individual tiny structures that you see here, which I refer to as the pebbles. Attached to it, you can see the black markings right here, you can see them here. What do you have attached to them? Well, you have your, on the outer part, you have most of it your B lymphocytes. Right? As you move from the outer all the way to the inner, then you have your deep cortex, and then you have attached to those, you have the T lymphocytes. So as the lymph flows through, you're going to have exposure of these lymphocytes into the new possible antigens that are trying to affect the body and trying to come back into your circulatory system. So it keeps moving down into the actual center of the lymph node, and this is known as the medulla, right? And that medullary portion, that one has B cells, right? As well as plasma cells, right? Plasma cells are the B cells that are found in the plasma. So B cells and or plasma cells. So once they move and they're exposed to both the T's and the B's, I mean all of them, then they're going to keep moving up. Hopefully by the time they get out of here, there is going to be what? No antigen on the next lymph node. So far so good? If there is an antigen on these lymph nodes, what's going to happen? What's going to happen if they have an antigen on these lymph nodes? A siren will sound, and then you're going to have a lot of what? Blood flow to what? To the area, and we call that inflammation. And that's when your lymph nodes is going to get what? Really big, inflamed, so that way you can get even more T's and more B's to fight the infection, right? As you expose the B cells, right, to these antigens, then the B cells will either bind to it, especially what B cell would you willing to bind to it? What is the one that you want? IgM, right? If it's a bacteria antigen, you want IgM to be, right? Or if it's a viral, then most likely will be a T cell, at least hopefully a TH, right? A T helper. Why we're not talking about killers? Because hopefully by the time that antigen got into your body was either through the skin or one of your mucosal glands, correct? And at that point, that's where your T killers come into play, right? So again, both T's and B's. So there's an inflammatory process. If there's an antigen going to be here. And that's where you're going to have something known as lymphadenitis, right? Inflammation of the lymph nodes. So here are the different parts of the lymph nodes, right? You have the outer part, which is a capsule. Then you have a subcapsular space. You have your outer cortex, which mainly was made out of what type of cells? What type of cells are found here? B cells, right? And then within the inner cortex, what do you have? Well, even though it's not here, but T cells, right? And in the germinal centers, what do you have? The B cells are plasma cells, right? B cells are plasma cells. Now, please do understand, these are all living tissues, so it requires to have what? Capillaries, right? So what gives that inflammatory process? Well, whenever there's an inflammatory process, there's what's going to happen? What's going to happen to the capillaries? The capillaries will dilate, and then there's more blood flow going to the area, right? That means there is going to be now increase in inflammation, correct? So every time you have a tissue that is inflamed, what do you have? Rubor, calor, dolor, all right, and function lyasa. Means ceases function, stop function. These are actually the cardinal signs of what? Of inflammation. So every time you have inflammatory process, these are the cardinal signs, right? That comes with it. And here is the order of the lymph flow. So number one, afferent vessels. So lymph comes in again through these pebbles that are in there. You have attached B cells, right, on the outer cortex, and from there it goes to the subcapsular space, right, when you have other type of cells as dendritic and macrophages. Okay, so what is a macrophage? Macrophage, you're going to see me drawing something like this, kind of like a Pac-Man, right? So every time there is a cellular debris, the macrophage is going to do what? Is going to ingest it through a process known of phagocytosis, right? So that's what the macrophages are. Now you have different type of macrophages, right? Dendritics is a type of macrophage as well. So it depends on where they are. They have different names. So as that moves all the way toward now the inner cortex, all the way to the inner cortex, we have exposure to T cells, and then from there it moves to the medulla, like mentioned before. And in medulla you have, of course, your B cells and or plasma cells, which is the one that's going to give you what? Humoral immunity. And there are two types of immunities, right? There's cell-mediated and there is humoral immunity. A cell-mediated is given by what? A direct contact with the cell. That's what we call it. Cell immunity. So a cell-directed immunity is the contact with the cell. Who does a direct contact with the cell? T cells, right? Cell immunity. What specifically type of T cell? Your NK cells, right? Your natural killer cells. The humoral immunity, humoral means what? Humor. Humor is actually a fluid from the body, right? So that means that it's going to be found within your plasma, it's going to be found within your lymph. So in there you're going to have your plasma cells, right? Your B cells. So that's why humoral immunity is mediated by the B cells. So which one is faster to stop the antigen? A cell or humoral? Cell, because it binds to the cell and kills it. Humoral, you have to get exposed, create an antigen, create an antibody for the specific antigen, and then create memories of it. Is that correct? So one will take within minutes, seconds to minutes, and the other will take days to weeks. Okay? And then finally, of course, after there is no inflammation, there was no antigen, or everything was cleared, then all the lymph is going to escape the hilum through the efferent vessels all the way to either right or left thoracic duct, right? Which then is going to drain at the level of the superior vena cava, right, into the right atrium. Natural killers are cell mediated. T cells, natural killers, cell mediated. So lymph node functions, right? What is the main function? 99%, they remove what? Cell debris. And in that debris is not only the parts of the cell that no longer are needed, but is what? Bacteria, virus, and fungi. So bacteria, virus, and fungi. Okay, so if you're a bacterial cell, who's going to mediate you? B cell. If you're a virus, who's going to mediate you? If you're a fungi, who's going to mediate you? B cells, right? So, of course, they provide, early warning. You want something early. that there's an infection in your body before it's already inside your heart and it starts eating your balls, right? And your endocardium. So that's why you have the multiple lymph nodes across your whole body. So the first step in immune response is to do what? Get the antigens, right? Make sure that you have no antigens in your heart. Once that lymph reaches your heart and mixes with the blood, you want to make sure that there's no antigens, because you don't want an inflammatory process in your heart, right? Inflammation process in your heart is devastating. So thymus, did we talk about the thymus already? Yeah, we said the thymus is actually a tissue, right? And then in the thymus, what was the only important thing about the thymus? The selection, right? So selection of whom? B's or T's? T cells, right? So T cells or T lymphocytes are the ones that are going to mature the thymus. So thymus has two thymic lobes, one on the right, one on the left. And you have a septa, which actually divides both of them, right? Divides each one of your lobes. Now, inside the thymus, all right, so we're looking at now the histology part of it. So in the histology, there is a medulla part of the thymus, as well as there is a cortex. And of course, septa is the divisions between each individual ones. Now, in the cortex, what do we have? We have dividing lymphocytes. Where do they become immunocompetent or mature? These T lymphocytes in the medulla. So the only thing that I'm adding to what you already know is the location, right? Immunocompetency takes place where? Cortex or medulla? Medulla, right? You divide them, you make more of the lymphocytes if needed, but they all need to go through the boot camp, right? Which is the immunocompetency. What is the hormone that allows you to make them immunocompetent or to stimulate the production of new lymphocytes going into this immunocompetency process, also known as the boot camp? Thymosin. And it's straightforward, right? Thymus releases what? Thymosin, which again allows for that lymphocytes to migrate and to actually go all the way into the medulla, as well as create more of them. So mature T cells, they leave the thymus, right? And from the medullary way all the way to your blood vessels, and now they're already in your bloodstream. These are immunocompetent. That means they are already differentiated, right? So a differentiated tissue or differentiated lymphocytes is to know what they need to do, right? They are immunocompetent. So let's go to the wording. Immunocompetent is they are differentiated, right? Means that they are mature. So if you see any of these three words, immunocompetent, differentiated, or mature T cells, means that they're all supposedly are working properly, right? Recognizing your own cells and killing only the antigens. That's the only positive selection. So there's another tissue which is known as the spleen. The spleen, we kind of talked about the spleen already, right? A couple of times. The spleen is a major organ that stores your red blood cells, right? The spleen is another place that we saw that red blood cells, as they get old, right? They went there and they're going to get killed by your macrophages. So apart from storing a storage of blood, the spleen also works as the following. You have within the spleen a white pulp and a red pulp. The white pulp is to do what? For lymphocytic, right, production. Again, most of your spleen is for a storage of what? Red blood cells. But on the red pulp, you're able to do what? You can have high amounts of storage, but at the same time, you're able to make more red blood cells. Remember when we talked about blood, we said that in fetuses, since they don't have long bones, or the long bones are not completely ossified, you need to depend on something else, and that was what? The spleen. So in your body, in an adult body, if your bones are, they have any problem, right? Being able to create red blood cells, so that new formation of red blood cells can also take place on the spleen. Okay? Same thing happens with the white pulp. Most of the lymphocytes are not created in the white pulp of the spleen, right? Most of them are found there. There's another place that they can hang, right? But they're able to be created from there too as well. Okay? But it's not a normal condition. So removal of abnormal and worn out red blood cells, storage of iron, and of course red blood cells, and initiation of immune response for both B's and T cells. All right, so it's another way of having them around the body. So that is pretty much the end of your lymphocyte. Your lymphocytes. So your lymphatic system, right? Part 2 of the lymphatic system. Now we're going to move all the way to the immune system. So on immunity, and actually we kind of went off and on immunity because both of them are related. They're not independent. Again, what is your immunity? Your white blood cells that are found where? Throughout your body, especially where? On your lymph nodes, in your blood, right? In your blood plasma. So we're gonna talk about basically for immunity, part 1, we're gonna talk about the difference between a specific and non-specific immunity. We're going to talk about pretty much a list of the non-specific defenses, and we're also going to talk about the humoral and the cell-mediated immunity, which is also known as cellular immunity, like the one that we just covered. So on the immune system, and this is actually a summary for the whole chapter. If you're able to grasp this, then it's a pretty nice summary of everything. So, in your body, you have a non-specific defense and what? Specific defense, right? What means non-specific? Means that you don't create anything specifically for that antigen, right? You're not making anything just for that antigen. You're making a structure, or you already have a structure that is capable of withstanding any damage or any antigens that may come forth into your body. So, a non-specific defense means that defense is innate. Innate means what? What does innate means? Innate, that is natural, that is you're born with it, that is part of you. So once you come out of that vaginal canal, these membranes or these innate defenses should be already in there, presence, okay, or almost completely developed. So mainly when you're born, what are you born with? You're born with surface barriers. Like what? Your skin. Are you born with a stomach? Hopefully yes. Within the stomach, do you have acid? So your pH of the stomach is about what? About 1, right? Are you born with cilia? Are you born with a respiratory tract? Yes. Does your respiratory tract have little hairs-like structures? Cilia. Yes. You're born with these things. Okay? Are you born with the capacity of, well, are you born with a tear gland? A lacrimal gland, you are, right? So are you born with the capacity of creating tears? Yes. So that's another kind of barrier, right? Okay. Now, as well as what? These are just the surface defenses. So you have defenses that are actually internals, right? And internal, not like the stomach, but even deeper, which is in your blood. Once you're born, mommy gives you something, right? What does mom gives you? By giving breast milk to you. Mom gives you IG. Mommy gives you IgG. Right? So IgG, it comes from mom. Remember that IgM was the first time you had a bacterial infection, right? Well, do you think, or you want your baby to have the first time bacterial infection? No, as a mom, you always want to do what? Protect it, right? So that breast milk that you're giving to your baby, it has the immunity that you acquired through your life. And that is found in the colostrum on breast milk. That means that baby has what? Immunoglobulins, right? B cells. They're not theirs, but at least they're borrowed for about what? You borrow them for about 3 to 6 months. Time enough for the baby to start creating this own stuff. Okay? 3 to 6 months. Now, are you born with phagocytes? You're born with macrophages. Yes. Are you able to generate fever when you're born? Hopefully, yes. Are you able, well, not you're able, do you have natural killer cells? Yes, it's part of your T's, right? Do you have antimicrobial proteins? Yes. Are you able to create inflammation? Yes. So these defenses are what? Your innate, correct? So far so good? That means, to make it clear, all these phagocytes, natural killer cells, that means you're born with what? With your white blood cells. I mean, they're not all mature. That's why mom has to give you the IgGs, but they're there. And little by little, they will become better. Now, that means you have what? Your phagocytic cells. What are some of the phagocytic cells? Your natural killers, dendritic cells, macrophages, neutrophils, eosinophils, basophils. What are these? So, is that specific? No, it's not specific. You're born with that. The surface plus as well whatever's in your blood, right? Or your white blood cells. Yes? Now, the specific defenses means what? That your body creates some structure or some molecular structure, protein, to bind to a specific what? Antigen. So that takes what? Time. That takes time. Because whenever you create something custom, it takes time. So that is known as what? Adaptive defenses. Adaptive means you have to do what to life? Adapt. If the body or your white blood cells or you as an individual do not adapt to the environment that you live in, what's going to happen to you? You will not survive, right? You will perish. Means what? There is a way of adapting of your body. It's either one through humoral and the other one through cellular immunity. What is humoral? Humoral means what? The creation of B cells. So this takes, again, days to weeks, correct? Now, what is the humoral? The D game. We talked about this already, right? Humoral means creation of your immunoglobulins, right? These are all your IgGs. So these are proteins, are part of your immune system, right? Like IgD, IgG, IgA, IgM, and IgE. As covered before. Cellular immunity. A cellular immunity means what? That you bind directly to your cells. And most likely, that uses what? Your T cells. What are your T cells? T helpers 1 and 2. T what? TK. What else? T suppressors and T cytotoxic cells, right? These are cellular immunity. These ones, the NK cells, are natural killer cells. These guys work as what? Look at it, because we covered before. Right? Even though we call it within the T's, right? They are a non-specific. Why? Because you're born with them. And born with them in the sense that they are able and capable, fully capable of doing what? Binding to an antigen. They don't know what that is. They're not specific for any antigen. As long as they recognize that it doesn't resemble their body or your body, it's going to automatically kill it. You're not making specific natural killers to kill something else. Is that clear? That's why they are non-specific. Is that clear? The T helpers, you make one specifically to recognize each individual antigen. The T suppressors, the same thing. And the T cytotoxics, the same thing. You need to make a specific one. Okay? So natural killers are, yes, they are some of your T cells, but they are part of a what? Innate defense, right? They're non-specific. So these guys, whether the B's or the T's, they're what? Part of your specific defense. Do you guys see it? So specific defense is all this right here. So within there, what are we going to have? Well, all the B's. So you have your plasma cells, your memory cells, your regulatory cells, right? On the T end, you're going to have your helpers, suppressors, cytotoxics, and the natural killers. But remember that we said the natural killers are non-specific, right? On that sense. That's the exception. So, the lymphoid system and body defenses. What are they talking about? Same thing. You have a non-specific defense, but this is just listed. Again, you're born with physical barriers. You're born with your phagocytes, which are your neutrophils and monocytes mostly. And neutrophils is actually the first line of defense. Right? Neutrophils is the first line of defense. Means whenever there is an antigen trying to enter your body through a cut, through one of your mucosal barriers, right? The first guys who are going to be there to neutralize the infection will be what? Neutrophils. Neutrophils neutralize. First line. And then you're going to have number 3, which is immunosurveillance cells. And these are your surveillance cells, your natural killer cells. Again, they are part of the same line of defense, but these are kind of like the second, right? Because first ones were neutrophils, followed by natural killers, and then macrophages. So that'll be the proper order. First, you send the neutrophils, try to see if they can accommodate and kill a couple things. If the infection is so huge, can neutrophils do it by themselves? No. So who do you gotta call? You call the natural killer cells because they are surveillance seeing the whole body. So they are around. You just need to send some signals, right? A phone call, which the phone call is now going to be called interleukins, right? Some signals, and these signals will call the natural killer cells to that site so they can help kill with the neutrophils. So far so good? Now, what do you have now? Since 1, neutrophils are killing, 2, and natural killer cells are killing. What do you have now? You're killing bacteria, so what do you have now? You have a lot of cellular debris, correct? So this is cellular debris. All right? Now, in that cellular debris, who's going to clean the cellular debris? Macrophages are going to come and try to clean that cellular debris. Does that make sense? So far, so good? All right. Now, can macrophages clean all the cellular debris? And if you think about it, I always paint the macrophages bigger than the cellular debris, right? Well, sometimes your cellular debris is huge and your macrophage is kind of tiny, like something like this. This is your cellular debris and this is your macrophage. Can that macrophage kill this cell debris? No. So, what does it need? Help, right? So, in order to need more help, it cannot do it by itself, but we did something. We said that there's a specific thing in our body, in our plasma, that coats the antigens on the outside, and they put like pom-poms or like phosphorescent lights around the antigens, which tells your body that's not part of you. And that was known as what? So, That was complement. So the complement system is just going to tag a bunch of proteins on the surface of the antigen, so make it more relevant or more seeable, right, to the immune system. So complement in conjunction with your macrophages, now they're capable of doing what? Destroying the cell or the antigen better. Now, can one macrophages clean everything? No. If you have 100 macrophages and you cannot clean everything, what do? You call more macrophages until you're able to clean it. So far, so good? Hopefully, within the macrophages, you have sufficient amount of digestive enzymes that you can destroy every single antigen there is and is invading your tissue. And nothing happened. You had a cut and nothing happened. Now, if you have too much of an infective organism or an antigen, what are the macrophages are going to do? Number 5, the macrophages will do what? They will present, that's a little flag, they will present the antigen to their surface. Means what? They're going to be called now antigen presenting cells, APCs. Why are we presenting something? It's really simple. I give you a challenge. The challenge is to eat a whole family pizza. It's about 24 inches. Can you eat it? No. And I said, you asked me, can I ask for help? And I'll say yes. So if you cannot eat it, what do? You take the piece of pizza out and say, hey guys, who wants to eat it? Right? So you're presenting the pizza, or in this case, the antigen, to other cells around it so they can help you do what? Digest it, because it's too much, you're already bloated. Or you're already full. Right? Make sense? So that's what we call antigen presenting cells. At that moment, as they flow through the bloodstream, they're going to encounter what? Other white blood cells, other T cells, other B cells, and they will interact between each other. So any of them, any white blood cells can help the macrophages. So far, so good? Complement just binds to the antigen and makes it more feasible for the body to recognize that that's an antigen. That's as far as you need to know right now. Again, this is the second slide. This is just an introduction. We're going to talk about each one of that more specifically, but that's the whole flow of events. If you grab it, you're in pretty good shape. I'm going to talk about it in more detail, guys. So let me just keep going. All right, there's more details to it. All right. So next one is you have interferons, and interferons, they help, but they actually help with bioreplication. Interferons interfere with bioreplication. These, you're born with that. Okay, you're born with that. That's part of your innate. So interferons interfere with what? With bioreplication. That means you have a virus and it's trying to get into your cell, and it prevents the virus to do what? To replicate his content within the cell. So that's what the interferons do. The next one is the complement. We talked about complement already, right? We said complement, just basically understand that is protein structures, right, that bind to the outer surface of a bacteria or an antigen. It makes it more recognizable for the immune system. Inflammatory response, think about it. If you ask for help, how are you going to get that help from point A to point B? So from your spleen to, I don't know, your intestines. You need to make more blood flow. So that's the inflammatory process, right? Vasodilation. And what produces this inflammatory process and vasodilation? Whenever you are presenting an antigen to a basophil, right? What happens? The basophil releases what? Oh, basophils creates and release histamine. What is histamine? It's an inflammatory, right, response mediator. So whenever there's histamines, it produces a vasodilation, so there's more blood flow to that area. Okay? And finally, we have the 5 cardinal signs, right, of inflammation. Remember? Rubor, which is redness. Dolor, which means what? Pain. What else? Tumor, which is a mass of tissue, a papule. And then finally, when that compresses everything, nothing comes in, nothing comes out. So we call it function lyasa, cease of function, right? So it gets to a point that there's so much inflammation that your body cannot get anything in or anything out, okay? So the tissue dies within. So you have your specific defense, which we talked about that already, right? So specific defense was what? That you create something or a cell specifically for that antigen. That was either what? T's or B's, right? That's what we talked here, guys. So what is non-specific defense? Now we're going to go in detail on each one. That was just the introduction. So a non-specific defense, we're going to talk about the skin, the mucous membrane, and of course the internal defenses. So step 1, physical barriers. You have your skin, which is stratified squamous epithelium, and hopefully that protects against a laceration all the way to your skin. So once that happens, then you have what inside your skin already. You have different type of what? Phagocytes. Phagocytes remove what? Debris and pathogens. Of course, you have different type of cells that can be a phagocytes. Okay? Especially what? Your macrophages and or your monocytes. So what is the difference between a macrophage and a monocyte? A monocyte is found where? Guys, there's a bunch of macrophages. They just change the name based on where they are located. So if your name here is John, when you go to Europe, you are Dr. Steve. They change the name. Why they do that? I don't know. They change based on but the role is the same. So a macrophage is found on your tissues. A monocyte is found on your bloodstream, just on the pure location. You also have dendritic cells, right? Kupffer cells. We're gonna talk about that later, okay? But remember that you have different type of cells, names, that they have the same function based on where they're located. So the next one is immunological surveillance cells, right? And we said that who is the immunological surveillance cells? Your NK cells, right? Which have non-specific defense. What do? They just bind to what? They bind to a cell that has been attacked by an antigen or is presenting an antigen. In this case, right? It destroys any abnormal cells. So if that is a natural killer cell, right, is going to bind to a macrophage that is doing what? Well, the macrophage couldn't eat the antigen, so now it's doing what? Presenting it. So guess what it's going to do? Kill the macrophage and kill the antigen. Because natural killers, what do? They kill. They don't know anything else. They're going to say, okay, let me help you. Let me take the antigen. Let me take it away from you. No, natural killers, they kill. And then they ask for your ID, right? They don't care if it's part of your body or not. As long as they know that there's some sort of antigen or you are weird or that cell structure is changed, in this case, not the cell structure, we'll say the molecular structure on the surface of the cell, right, has changed, then automatically tells the natural killers to eliminate that cell. Okay. Number 3, if it finds that there is number 2, my bad. Number 2, so then you have your interference, which is what we talked about. And again, interference is basically for bioreplication. So they prevent virus from infecting or keep infecting other tissues or other cells. Then you have your complement system. Again, the complement system, are you born with these things? Yes, you're born with these things. So the complement system is going to attack break down the what? The cell wall, okay? And it's going to promote an inflammatory process. You can see these are the complement proteins, and they're bonding to what? To the surface of these capsular bacteria. What are they doing? They're sending signals to stimulate what? Inflammation. And they're going to promote as well what? Immune response. Remember? So, if that is all good, then at the end, what are you going to have? Lysis, right? You're going to kill that bacteria. Number 3, right? You create an inflammatory response because this dictates the inflammatory response. Inflammation, it has multiple effects in your body. What are these multiple effects? Well, they're listed here. So, increase in blood flow, yes? Phagocytes are activated. Because the more blood flow, as phagocytes reaches there, there's going to be more exposure to the antigens. Are you going to increase the permeability of the capillary? What does that even mean? That means, are you going to allow the phagocytes to go in between that blood vessel? Remember, in the bloodstream, they're called what? Monocyte. Do you want them in the bloodstream? No, the infection is where? In your tissues. So they have to get out of the bloodstream into the what? The tissue. And from the tissue into the bloodstream, back and forth. So you are required to increase what? Capillary permeability. By increasing capillary permeability, you're increasing the flow of white blood cells, right, into the tissue itself. So you can help, right, the invasion that is in your body already. Complement will be activated because as you release inflammatory chemicals, right, from the mast cells, the complement will be activated. You're also going to do what? Clotting. But this is actually walls off. What it means? What does that mean? So if my inflammation is around this area, would you like these cells or this infective organism to migrate anywhere in my body? No. What are you going to do? You're going to send clotting factors, and these clotting factors will start to do what? To the infective tissue or area. What am I doing? I'm creating a what now? A barrier. I'm creating a wall. And that's what they do. They wall off. Means things can come in, but nothing can do what? Come out. Why do we do that? To prevent from the infective organism or antigen to spread through the nearby tissue or cells. Next one. As that inflammation occurs and more blood flow goes to the tissue, what do you have? The temperature increases, right? On that area, the temperature increases. And then you are going to now, see, that's the last part. You're going to activate what? Your specific defense. Why is that the last thing you do? Because specific defense requires what? Time. Okay? That means what? You first are going to use all your neutrophils, your natural killers, all your defenses that you already have. And while that is happening, what are you doing? You are stimulating your specific defenses. Make sense? So far so good? To create now what? To create memory, either to cell mediated or humoral mediated. You're creating memories of T cells and memories of B cells. That's what you're doing. And of course, finally, you have your body temperatures. But what is the one that increases that overall body temperature? So this increase on temperature that we saw here is only on the area. If the infection keeps spreading and that becomes part of your blood, then that means you have a sepsis, right? Because the infective organism went from the tissue into the blood. So now you have a response of your body by stimulation of pyrogens. And pyrogens, what they do is they increase overall your body temperatures. Okay? So this is the order of events. First you have your neutrophils and your NK cells that we mentioned. Then you have your phagocytes, eosinophils, and cytotoxic T cells, right? And then finally, you make what? Your specific defense, number 7. And that specific defense is the one that we see here. Plasma cells, B cells, right? So far so good? So that is the order, right, of how your body deals with a antigen as it gets exposed. Here's a chart just to prove that fact, that what? These are the actual immune cells, and this is the time in weeks. Which is the first one? Neutrophils. What's the next one? Natural killer cells. Which is the other one? Cytotoxic T cells, and then macrophages, right? And so forth. So it's telling you, right, the order that these things occurs. Okay, and then for the next class, we're gonna talk about each one of them, right? The physical barriers and each individual cells. So for today, that's it for the first part of the immune system.